Hello everyone, I am Sarita Rawat, Assistant Professor, School of Pharmacy, Suresh Gyan Vihar University, Jaipur, Rajasthan. Today is our next lecture of Pharmaceutical Microbiology. Today's topic is Structure of Bacterial Cell. So, learning objective of today's lecture is to study about size of bacterial cell, to study about shape of bacterial cell, to study about various structure part of bacterial cell. So, content of today's lecture included introductions, cell morphology, structure of bacterial cell, which includes cell wall, plasma membrane, extracellular external structure, and intracellular internal structure. Now, learning objective of today's lecture include you are able to know about upon the completions of this lecture size of bacterial cell, shape of bacterial cell, various structure parts of bacterial cell, and comparison of prokaryotic versus eukaryotic cells. Now, introductions of today's lecture include we will start with the basic introductions of the cell, bacterial cell. So all bacteria are unicellular organisms that reproduce by binary fissions, and most bacteria are capable of independent metabolic existence and growth, but species of Clementicia and Rickettsia are obligatory intracellular organisms. So bacterial cells are extremely small and are most conveniently measured in microns, and bacterial cells are very usually between 0.4 and 1.5 micrometer in short diameters. So when we talk about the structure of bacteria, they are smaller and simpler in structure than the eukaryotic cell, with no recognizable organelles, and all the activities performed by the organelles also take place in the bacteria, but they are not carried out by any specialized structure. So the small size, simple design, and broad metabolic capabilities of bacteria allow them to grow and divide very rapidly and to inhibit and flourish in almost every environment. Now, structure of bacteria, they were first seen under a microscope by Anton van Leeuwenhoek in 1676, and as microscopes have improved, scientists have came to understand the bacterial cell structure better. Now, as you can see, this is the simple structure of bacterial cell. So it includes chromosome, phylus, FIMBRIAE, ribosomes, and inoculation, flagellum, plasmid, cytoplasm, cell membrane, cell wall, and cellulose capsule or slime layer. Now, coming to the basic point, bacterial cell structure. So it is organized into three categories: internal structure, which includes cytoplasm, nucleoid, bacterial chromosome, plasmid, ribosomes, and storage granules. Then the second, cell envelope. So cell envelope include cell membrane, peptidoglycan, cell wall, or an outer lipid membrane only found in the gram-negative cells. So, and the another part is external structure. So external structure include flagella, FIMBRIAE, sex phylus, and glycols. Now, when we talk about the intracellular structure of a bacterial cell, so it includes cytoplasm, chromosome, plasmid, ribosomes, inoculations, bodies. So when we talk about the deep study of intracellular organ, so cytoplasm. So cytoplasm is protein of the cell that lie within the plasma membrane, and substance within the plasma membrane excluding the genetic material. They have gel-like matrix composed of mostly water, 4 divided by 5th, enzymes, nutrients, wastage, and gases. And they contain cell structure, ribosomes, chromosomes, and plasmid, as well as components necessary for the bacterial metabolism, and it is relatively featureless by the electron microscopic, although small granules can be seen, and carries out a very important functions for the cell, that is growth, metabolism and replication. So that is the function of cytoplasm. Now coming to the constituent of cytoplasm, it include protein including enzymes, vitamins, ions, nucleic acid and their precursors, amino acid and their precursors, sugar, carbohydrate and their derivatives, fatty acid and their derivatives. Now nucleoid, coming to the nucleoid, it is unlike the eukaryotic true cells, bacteria do not have a membrane enclosed nucleus. So the nucleoid is a region of cytoplasm where the chromosomal DNA is located. So it is not a membrane bound nucleus but simply an area of the cytoplasm where the strands of DNA are found. Now plasmid. Plasmid is a small extracellular extrachromosomal DNA, and it contains genesis for antibiotic resistance or virulence, and structure similar to the most bacterial chromosomes but considerably smaller. Now plasmids are conventionally closed circular DNA, and in a few species linear plasmid have been found. So size, when we talk about its size, so chromosomal DNA is typically about 4000 kb and plasmid DNA ranges from 1 to 200 kb. Now number of plasmid included 1 to 700 copies of plasmid in a cell. When we talk about its function, so the function of plasmid is not well known, always known, but they are not normally essential for survival of host, although their presence generally give the whole host some average and antibiotic resistance. So some plasmids code for proteins that degrades antibodies, a big advantage for pathogens, and some encodes for the proteins which confer virulence factor on the host, and for example E. coli plasmid ENT P307 code for an enterotoxin which makes E. coli pathogenic. Now conjugated plasmids, so these allow exchange of DNA between bacterial cells. So plasmid are the associated trends can be transferred between bacteria, even from one bacteria species to another. Plasmid are not involved in the reproduction process. Plasmid replace independently of the chromosomes, and plasmid are passed to other bacteria by two means. So for most plasmid types, copies in the cytoplasm are passed to the daughter cells during the binary fissions. Now plasmids, other type of plasmids from tube-like structure at the specific space called a phylus that passes copies of the plasmid to other bacteria during conjugations, a process by which bacteria exchange genetic informations. Now plasmid have been shown to be instrumental in the transmission of special properties such as antibiotic drug resistance, resistance to the heavy metals, and virulence factor necessary for the infection of the animal or plant hosts. So the ability to insert specific genes into plasmid have made them extremely useful tool in the area of genetic engineering and rDNA technologies. So ribosomes-protein synthesis machinery. So how it works? It consists of RNA and protein. It is abundant in cytoplasm. It has often grouped in long chain called polyribosomes. and it gives the cytoplasm of bacteria a granular appearance in the AM. And similar than the ribosomes in electrolytic cell but have a similar function, and bacterial ribosomes have sedimentation rate of 17 as their subunit have rate of 30s and 50s. So the unit used to measure the sedimentation velocity is Safeburg. So as you can see, this is the smallest subunit and this is the large subunit. Now ribosomes, what are ribosomes? So they translate the genetic code from the molecular language to of the nucleic acid to that of the amino acid, the building blockers of the protein. Bacterial ribosomes are similar to those of eukaryotic but are smaller and have a slightly different composition and molecular structure. So bacterial ribosomes are never bound to other organelles as they sometimes are bound to the endoplasmic reticulum in eukaryotes but are freestanding structures distributed throughout the cytoplasm. And there are sufficient differences between bacterial ribosomes and eukaryotic ribosomes that some antibiotics will inhibit the functioning of the bacterial ribosomes but not eukaryotic. So, thus killing bacteria but not the eukaryotic organism they are infecting. Now, streptomycin bind with the 70s ribosomes and stop the protein synthesis, but it cannot bind with 80s ribosomes of eukaryotics, and then thereby eukaryotic cell remains unaffected by the streptomycins. Now, what is bacterial chromosome xenophore? So, the bacterial chromosomes consist of a single circle of deoxyribonucleic acid, and DNA is double standard, two standard line up anti-parallel to each other, and the base are linked together with hydrogen bonds, and it includes most of the genetic material of the organism. So, inclusion bodies, bacteria can have their within their cytoplasm a variety of small bodies collectively referred to as inclusion bodies, and some are called granules and other are called vessels. Now, inclusions are considered to be non-living compound of the cell and that do not possess the metabolic activity and are not bounded by membranes. And the most common inclusions are glycogen, lipid droplet, crystals and pigments. So, when we talk about the granules, inclusion bodies, so granules are densely compacted substance which are without a membrane covering, and nutrients and reserves may be stored in the cytoplasm in the form of glycogen, lipid, phospholipid, or in some cases sulfur or nitrogen for the later use. And each granules contain specific substance such as glycogen, glucose polymer and phosphate. Phosphate polymer supplies energy to the metabolic process, and sulfur bacteria contains reserve granules of sulfur, and these granules are depleted in the starvation process. Now, inclusion bodies, vessels that are some adequate phosphosynthesis bacteria and cyanobacteria have rigid gas-filled vacuoles, and it helps in the floating at a certain level, allowing them to move up or down into the water layer with different light intensities and nutrients level. And some magnotate bacterium the presence of such magnetic inclusions enables these bacteria to respond to the magnetic field. For example, ferric oxide that we also call stores magnetates and then this is all about your inclusion body vessels. Now next is our micro compartment. So what is micro compartment? So, microcompartments are widespread membrane that are bound organelles that are made of a protein cell that surrounds and enclose the various enzymes. And next we have carboxysomes. So, carboxysomes are protein enclosed bacterial microcompartment that contain the enzyme that involved in the carbon fixations. Now, then we have a magnetosomes. So, magnetosomes are the bacterial microcompartments which are present in the magneted bacteria that contain the magnet crystals. Now, what is cell envelope and what is it contain? So cell envelope mainly contain the plasma membrane, the plasmic space, and the cell wall and the outer membrane. Now, plasma membrane include phospholipid bilayer surrounding the cytoplasm and regulates the flow of substance in and out of the cell, and it consists of both lipid and protein. So it protects the cell from its surrounding and selectivity, permeability to the ion and organic molecules, and control the movement of substance in and out. So, numerous proteins moving within or upon this layer are primarily responsible for the transport of ion, nutrients, and waste across the membrane. Now the next we have periplasmic space. So periplasmic space basically include the gram-negative bacteria. In gram-negative bacteria, the space between the cytoplasmic membrane and the cell wall, and the space found between cell wall and the outer membrane is periplasmic membrane may constitute up to 40% of the total cell volume in G-positive species, and gram-positive bacteria space between the cytoplasmic membrane and the cell wall. So the periplasmic is filled with the water and protein and is mini sciences of the cytoplasm. Now, when we talk about the periplasmic space, so however, periplasm contains protein and other molecules distant from those in the cytoplasm because the membrane prevents the free exchange between these two compartments, and periplasmic proteins have various functions in the cellular process which include transport, degradations, and motility. Now, periplasmic controls molecular traffic and entering and leaving the cell. Now, outer coverage of the most cell that protect the bacterial cell and give it shape are included spherical shape, rod shape, and spiral shape. So they composed of peptidoglycan, polysaccharides plus protein. So microplasma are the bacteria that have no cell wall and therefore they have no definite shapes. Now, when talking about the cell wall, so peptidoglycan molecule found only in the bacterial cell wall, and the rigid structure of peptidoglycan give the bacterial cell shape, surround the plasma membrane, and provide prokaryotic with the protection from the environment. And from the peptidoglycan inwards, all bacterial cell are similar. And going further out, the bacteria world divides into two major classes that involve gram-positive and gram-negative bacteria. And amount the locations of peptidoglycan in the cell wall determines whether a bacterium is gram-positive or gram-negative. Now, peptidoglycan is a polysaccharide plus protein. So, peptidoglycan murine hues polymer of indoor locking chains that is composed of similar monomers. And peptidoglycan is made from polysaccharide chain that is cross-linked by peptides containing D amino acids, and the backbone of peptidoglycan's molecule is composed of two derivatives of glucose, and that is N-acetylglutamine, that is NAG, and N-acetylmuramic acid, so NAM. So the NAG and NAM stands for connected by the inner peptide bridges. Now, gram-positive cells. So, gram-positive bacteria possess thick cell wall containing many layers of peptidoglycan and trichoic acid. So, in gram-positive cell, peptidoglycan is the outermost structure and made up of as much as 90% of the thick, compact cell wall. Now, when we talk about the gram-negative bacteria, so gram-negative bacteria have relatively thin cell wall consisting of few layers of peptidoglycan that is surrounded by a second lipid membrane containing lipopolysaccharide and lipoproteins. And peptidoglycan make up only 5 to 20% of cell wall and in not the outermost layer, but lie between the plasma membrane and an outer membrane. And now when we talk about the external structure, so external structure of cell include flagella, pili or fimbrioid, and capsule and slime layer. Now when we talk about the flagella, so it is a singular flagellum and long white-like semi-rigid cylindrical structure that aids in cellular locomotions and function much like the propeller on the shape. Now about 20 nanometer in diameter and up to 20 micrometers in the length, and diameter of a prokaryotic flagellum is about 1/10 of that of the eukaryotic. So flagella are derived by the energy released by the transfer of ions down on electrochemical gradient across the cell membrane. Now, flagella is made up of protein subunits called flagellum, and each flagellum is attached to the cell membrane with the help of proteins other than flagellum, and the basal region has a hook-like structure and a complex basal body, and the basal body consists of a central rod or shaft surrounded by a set of rings. Now, flagella bacterial species differ in the number of arrangement of flagella on their surface. So, bacteria may have one, a few or many flagella in the different positions on the cell, and microtracheus is a single flagellum, and amphitracheus is a flagellum at each end. Lepotracheus is a cluster of flagella at the pole of the cell. Now, Patriarchus is a flagella distributed over the entire surface of the cell. Now, as you can see, this is the picture of the arrangement of flagella. So, you can see the first flagella, only one flagellum it has, that is called monotracheus, single flagella. Then the B we have, it is amphitracheus, a flagellum at each end. And then the C we have lipotracheus, you can see. And then we have the patritracheus flagella distributed over the entire surface. So, as you can see here. Now, motile bacteria are attracted or repelled by the certain stimuli in the behaviors called taxis. These included chemotaxis, phototaxis, and magnotaxis. So, the flagella wage in a propeller-like motions to help the bacterium move toward the nutrients and it away from the toxic chemicals toward the light, that is photosynthetic. Cyanobacteria and prokaryotic exhibit a variety of movements and move, swim, tumble, glide, swim in the response to environmental stimuli. Now, hollow hair-like structure structure made of proteins, so which involved in the attachment to solid surfaces or to other cell and are essential for the virulence of some bacterial pathogens. Now, fibrin is the fine filaments of protein just 2 to 10 nanometer in the diameter and up to several micrometer in length. Now they are distributed over the surface of the cell and resemble fine hairs when seen under the electron microscope. Now, fibrin and phyli. So, what is phyli? Phyli is a phylus, or single phyli is a phylus, are cellular appendages stately larger than the fimbriae, and it involved in the attachment to surface. And specialized phyli, the sex phyli, allow the transfer of genetic material from one bacteria to another, and in a process called conjugations, where they are called conjugations phyli or sex phyli. So, types fourth phyli is generate a movement and helps in the conjugations and pathogenicity. Now, bacterial reproduction. So, bacterial reproduction include cell growth and reproductions by cell division are tightly linked in the unicellular organism, and bacteria grow to a fixed size and then reproduce through the binary fission and a form of asexual reproduction. So, under optimal conditions, bacteria can grow and divide extremely rapidly, and bacterial population can double as quickly as every 9 to 8 minutes. So, in cell divisions, two identical clone daughter cells are produced. Budding involves a cell forming a proteasome and breaks away the process. A daughter cell produces a daughter cell. Now, bacterial growth. Growth of bacteria is the orderly increase of all the chemical constituent of the bacteria, multiplications in the consequences of growth, and death of bacteria in the reversible loss of ability to reproduce. Now, factors required for bacterial growth. So, the requirement for bacterial growth are environmental factor, source of metabolic energy. So, environmental factor affecting bacterial growth involve nutrient. So, nutrient in the growth media must contain all the elements that are necessary for the synthesis of new organisms. So, it includes hydrogen donators and acceptors, carbon sources, nitrogen sources, minerals, sulfur and phosphorus, growth factor, amino acid, purine, pyrimidines, vitamins, and trace animals, magnesium, ferrous, and temperature. Now, temperature include microorganisms are sensitive to temperature changes. So, usually unicellular enzyme have temperature optima. If temperature is too high and protein denaturate, including enzyme carriers and structure components. So, temperature ranges are enormous. It is in -20 to 100 degree Celsius. Now, temperature include organism exhibit the distance cardinal temperature, minimal, maximal, and optimal growth temperature. So, if an organism has a limited growth temperature range, so stenothermal, which we call stenothermal. So, for example, Angorei, and if an organism has a wide growth temperature range, so we call it eurothermal. So, for example, we have E. facilis. Now, when we talk about the temperature, so phascophils can grow well at 0 degrees Celsius and optimal growth at 15 degrees Celsius or lower, and usually will not grow above 20 degrees Celsius. Now, Arctic and Arctic oceans, Antarctic and Antarctic oceans, protein synthesis, enzymatic activity and transport systems have evolved to functions at low temperature. Cell walls contain high level of unsatisfied fatty acids, which include semi-fluid when cold. Now, Psychotropes can also grow in 0 degrees Celsius but have the growth optima between 20 degrees Celsius and 30 degrees Celsius, and the growth maxima at about 35 degrees Celsius. So many are responsible for food spoilage in refrigerators. So mesophils have growth minima of the 15 to 20 degrees Celsius, optima of 20 to 45 degrees Celsius, and maxima of about 45 degrees Celsius or lower. So majority of human pathogens have this. Now thermophils. So thermophils have growth maxima around 45 degrees Celsius and optima of 55 to 65 degrees Celsius. So now next we have hyperthermophils. So hyperthermophils have grown minima around 55 degrees Celsius and optima of 80 to 110 degrees Celsius, like in sea floor and sulfur vents. Now when we talk about the physical pH, so pH is the negative logarithm of the hydrogen ion concentration. So acidophils grow best between pH 0 and 5.5 and neutrophils grow best between pH 5.5 to 8.0. Alcophils grow best between pH 8.5 and 11.5. Now extreme alcophils grow best in the pH 10 or higher. Now sudden pH changes can inactivate the enzyme and damage the plasma membrane. So reason for buffering culture medium usually with a weak acid conjugate base pair, for example KH2PO4 K2HPO4 is a mono-base potassium dis-basic potassium. Now as you can see, these are some examples of the bacteria that grow in the minimum pH, optimum pH and maximum pH. So you can check them out, some prokaryotes. Now oxygen and growth. So these are some bacteria and then which grow in the aerobic environment and anaerobic environment and the effect of O2 on them. Now water availability. So water is solvent for biomolecules and its availability is critical for the critical for the cellular growth, but the ability of water depends upon its presence in the atmosphere, relative humidity, or its presence in the solution or substance that we call water activity. So water activity of pure water is 100%, is 1.0 affected by dissolved solute such as salts or sugar. So microorganisms live over a range of what we can say water activity from 1 to 7 1 to 0.7 and the AW of human blood is 0.99. so seawater is 0.98 and maple syrup is 0.90 and great salt lake is 0.75 and water activities in agriculture soils range between 0.9 to 1. Now, when we talk about the source of metabolic energy, so mainly three mechanisms generate metabolic energy. These are fermentations and respirations and photosynthesis. So, an organism to grow, at least one of these mechanisms must be used. So when we talk about the morphology of bacteria, so bacteria display a wide diversity of shape and size that we call morphologies. So it cannot be seen with the human eye, so we have to see them with microscope. So they are microscopic. So neurologist Antonio van Leeuwenhoek saw microscopic organism in a variety of substance with the aid of primitive microscope. So now bacteria are usually examined under the light microscope and capable of more than 1004 magnifications and details of their internal structure can be observed only with the aid of much more powerful transmission electron microscope. So unless special phase contrast microscope are used, bacteria have to be stained with a color dye so that they will stand out from their background. Now, when we talk about the size of bacteria, bacteria are the smallest living creatures, and most bacteria are 0.2 μm in the diameter and 2 to 8 μm in length. So, bacterial cells are about 1/10 the size of eukaryotic cells and are typically 0.5 to 5 micrometers in length. Now, giant bacteria, for example, Thymomarginata, so they are the up to half a nanometer long and are variable to the unaided eyes. So then we talk about among the smallest bacteria are the members of the genus Mycoplasma, which measures about 0.1 to 0.25 μm in diameter and as small as the largest virus. So some bacteria may be even smaller, but these ultra microbacteria are not well studied. So E. coli, a normal inhibitor of the intestinal tract of the human and animals is about 2 μm long and 0.5 μm in diameter. So spherical cells of Streptococcus aureus up to 1 μm in diameter. So now we can see Cyanobacterium are average in 6 μm by the 12 μm in size. Now, bacteria come in a wide variety of shapes. Coccus are spherical or oval shaped, and bacillus are round, inheaded, cylindrical shaped cells. Vibros are comma shapes, curved rods, and derive the name from their characteristics vibrity modality. So, spirals are the rigid spiral form like coli, and spirochetes are the long, slender, and flexible spiral form. Then we have filamentous also. They resembles radiating rays of sun. Then they are in generally characteristics of a given bacterial species but can vary depending on the growth conditions. And some bacteria have complex life cycles involving the production of stakes and appendages. Now, bacteria generally from distensive cell macrophages when examined by the light microscopy and direct colony morphographs when grown on the Petri plates. So when we talk about the cell shape, this wide variety of shapes is determined by the bacteria, cell wall and cytoskeleton. Shape of the cell is important because it can influence the ability of bacteria to acquire nutrients attached to the surface, swim through the lipids and escape predators. Now, as you can see, this is the arrangement of cell. Cellular arrangement occurs singularly in the pairs in the chain and in clusters. So, single cellular is called coccus. Then in pair called diplococcus. In 4 pair is called tetrad, and in chain it's called streptococcus, and in cluster it called staphylococcus. Now, bacilli, diplobacilli are 2 cells, and trizid are 4 cells, plazid are 2 cells arranged parallel, or streptobacilli change arrangement, for example, E. coli and Salmonella. So, as you can see, these are the roll-shaped bacilli and single bacilli, pair bacilli and chain bacilli. Now, as you can see, these are your cocci, these are the bacillus, single bacillus, diplobacillus, streptobacillus and coccobacillus. Now these are vibro and spirillum and spirochetes. Now the other shape we have a star-shaped bacteria, rectangular bacteria. Now, bacterial classification based on shape. So, it includes bacilli, road-shaped bacteria, coccus, spiral or oval-shaped bacteria, and spiral-shaped bacteria are called spirilla, and comma-shaped bacteria, for example, vibrio, and branching filamentous form we have actinomycete. Now, bacterial classification based on staining method. So, we have two kinds of method: gram-positive bacteria, gram-negative bacteria. So, gram-positive bacteria take up crystal violet dye and retain their blue color or violet color. Gram-negative bacteria do not take up crystal violet dye and thus appear red or pink. Now, classification based on the respiration. So, aerobic respiration. So, sugar are break down in the presence of oxygen to produce carbon dioxide, water and energy. Anaerobic respiration break down the sugar and release energy in the absence of oxygen. Anaerobic respiration is typically slower and lesser effect than the aerobic respiration. So, anaerobic respiration involve chemical and other than oxygen and carbon dioxide. Now, classification based on respiration. Next, we have facultative anaerobic respiration. So, facultative anaerobic are able to perform either aerobic, anaerobic respiration depending on the oxygen content of their environment. So, for example, coliform bacteria. Then we have microaerophils. So, they are sugar are broken down in the presence of minute amount of oxygen to produce the energy. Now, these are the classification based on environment. So, we have mesophils, neutrophils, enteromophils, acidophils, alkanophils. Then we have halophils and osmophils. Now, classification based on flagella. So, we have no flagella, uniflagella, biflagella and more flagella. Then classification based on spore formation. So, we have spore forming and non-spore forming bacteria. Then for nutritional source, classification based on nutritional source. So, bacteria are also classified based on the type of energy source utilized by them for the survival. So, autotrophs, photoautotrophs and chemoautotrophs. Now, nutritional source that are we have heterotrophs. So, heterotrophic bacteria obtain sugars from the environment and they are in the living cell and organisms they are in. So, symbiotic, sporadic and parasite. Now, bacteria are classified based on various factors. So, shape, cell wall structure, respirations, type of nutrients, nutritional source, characteristics, environmental factors, and many others. Now, coming to the reference part, we have taken all the references from Bacteria in Biology, Biotechnology and Medicine, 4th editions, www.structureofthebacterialcell.com, and then we have taken some from Pharmacological Microbiology Backwells, and then from the Pharmaceutical Microbiology by Dr. N.K. Jain. Then today we have concluded some quiz questions for you that are based on the lecture we have already studied. So you can easily answer these questions while going through all the lecture. So how does bacteria move? Live bacteria are found in milk, cheese or yogurt, and bacteria can only be spread through coughing. Bacteria are only found in the dirty places. The flu is caused by bacteria that help break down dead material in the environment are called. That the outer coverage of a bacteria cell is called. And then bacteria are prokaryotes, which means they and special chemicals that some bacteria produce to stop other bacteria from growing or killing it, what it is called. And bacteria cells are exactly like animal cells. So you have to answer them either true and false, or either you have to answer their particular question. So while you going through all the lecture, you will get to know about these questions. And for more informations, you can definitely follow these references. These are taken from some slide shares, pharmaceutical microbiology NKJ, and then we have taken from the Pariscott, LM, Harley, GP and Calil, DA Microbiology 3rd editions, which is Oxford publications, Brown publisher, Oxford. And then we have also followed the pharmaceutical microbiology by Backwell scientific publications for Oxford. So you can definitely check out these references for more knowledge and further knowledge. So you will enhance your knowledge by going through these references. So thank you for today's lecture. Thank you for kindly listening to me, and we have covered all the basics informations about bacteria, structure of bacteria, how we categorize the bacteria. So thank you for today's lecture.